Okay, thank you, Francisco. So yeah, great pleasure to be here. Always great to see familiar faces and present the recent research. I think it's a great platform, so great to see everybody. So I would like to talk about the project we are pursuing. Yeah. So we'd like to talk about the project we're pursuing in the labs, about the benchmarking of RNA-seq TCR profiling methods. So all the work done by Kari, who is the talented graduate student in the lab, and if you want, everything I will be talking about is available in the preprint. So some background to start with. TCR-seq as the technology provides estimates of the TCR repertoire to measure the effectiveness of immunotherapy and overall prognosis. However, the available TCR-seq data is limited comparable to RNA sequencing. So we're talking about several thousand samples available publicly in terms of TCR-seq data and multiple tens of thousands of RNA-seq data. And actually, the bioinformatics community was working hard to develop the method to profile the TCR repertoire from RNA-seq data. However, the relative performance of the tool is currently unknown, as well as the ability of RNA-seq-based tool to characterize the repertoire across different tissues currently unknown. So that's the motivation for the benchmarking. So I hope you're on the same page with me. That's always required to do benchmarking to assess relative performance of the bioinformatics tools. So just a little bit of background. So what we mean by the TCR repertoire. So if you think about the TCR repertoire, that is actually encoded by several loci, and the mechanism beyond the TCR repertoire is quite fascinating. So if you zoom in one of those loci, so you can see that there is a variety of genes called V genes, D genes, and the J genes, and then they can actually recombine in random fashion. However, so like that. So, however, that will be not enough to achieve the astonishing diversity we know of the TCR repertoire to be estimated 10 into the power 15, right? So we need some more mechanisms. So another mechanism, you're allowed to insert non-template insertion at the junction point, right? So that mechanism literally can generate any sequence, and that's why the diversity of the TCR repertoire is estimated in 10 into the power 15. So several definitions will be referring to several regions called complementary determining region, number 1, number 2, and number 3. And actually CDR3 is quite interesting because it harbors the most diversity. So we'll use that as the proxy to define the clonotype. And the collection of the clonotype of the particular individual we will refer as the repertoire. So what that means in terms of the reads, right? So that's unknown to us, right? So we don't know which combination occur and what was the insertion. So what we do know, we know the reads given by the sequencing platform. So fundamentally, there are two types of the reads which were mapped to the human genome and the ones which were unmapped to the human genome. So the mapped are present in the red color and unmapped are gray. So why they are unmapped? Because actually the V gene and the G gene and the J genes are part of your genome, right? So if the read is entirely mapped to the V gene, it will be mapped normally to the human reference. However, if the read is spanning the recombination, kind of overlapping that CDR3 region, that will be unmapped. So this is the reads we'll be focusing on. So I guess, again, the motivation for that the data coming from the proper technology, which we actually, by the way, will be using as the gold standard, called TCR-seq, is limited, right? So, and that's why it's not suitable technology to reconstruct the immunome, the complete set of the receptors of the particular individual. So, few more definitions before we jump on the results. So we'll talk about monoclonal and polyclonal repertoire. So what we mean by monoclonal, we define that as the repertoire with the immune diversity estimated by the Shannon entropy less than 2, and the polyclonal more than 2. So what that means practically, meaning that you have a certain clonotype to be dominating the repertoire, as you have here, the blue one, and on the right, you kind of don't have a dominant clonotype, and all the clonotypes are more or less have equal proportions. So this is the data sets we were able to assemble, where we do have RNA-seq on one hand and TCR-seq on another hand. So we're going to use TCR-seq just because it's a proper technology as the gold standard, and we will assess the performance of the tools to use RNA-seq to reconstruct the TCR repertoire. So two data sets right here on like PBMC and the skin samples are in-house data generated by our collaborators at UCLA, and others are the public data. So what you can see here, there is quite a variety in terms of the sample, in terms of the number of samples, in terms of the sequencing depth, as well as the read length. And what is important, there is quite different in terms of the fraction of the T cells in the tissue. So we're talking about T cell poor and T cell rich tissues. So tissues like skin don't have much T cells. However, PBMC will have a lot. We also have some proportions of the monoclonal versus polyclonal repertoire. So that gives us a good kind of benchmark data sets we can assess the performance of the tools. So this is how kind of we summarize that, right? So we have two criteria. Is it the tissue poor or rich? Is it monoclonal or polyclonal? And we have kind of more or less combination of all of those parameters. So in terms of the tools, we are aware of 3 tools being developed so far. One of them is our tool called IMRAP, we developed a while ago, and there is another one, Mixer and Trust 4. So there are 3 tools. As you can see, they vary in the way they were implemented, in terms of the language, in terms of the format they use, and some other characteristics. So we were curious to know what is the relative performance of those tools across different tissues. So the first results I would like to present is talking about ability to successfully capture the monoclonal repertoire. So the monoclonal repertoire here are both colors, right? So we are talking about rich tissues, which are blue, and the poor tissue, which are orange. So what you can see, so that's on the y-axis, that's a fraction of the repertoire being captured, calculated as the sum of relative abundances of the clonotype. And then x-axis tell you the relative threshold of the clonotype. So for example, here, if you look at the very left plot, so if all the clonotypes would be with the relative abundance, with certain relative abundance, you will capture all of them, right? So like that's what the line stands for. However, as more rare clonotypes you have got, as harder for an ISMB is to capture, one, due to limitations of the bioinformatics method, or just due to the very simple facts, you don't have reads coming from the rare clonotypes. So overall, we see consistent performance of all the tools where rich monoclonal tissues are much easier to capture the repertoire. As the tissues are poor, so there are not many reads kind of coming from the TCR, but they are monoclonal, you can do a decent job to capture the common clonotypes in opposed to rare ones. So as we move on to the polyclonal, things are much, much, much harder, where both in the rich and poor, I think for each you're getting some decent results. You're capturing again the rare, the highly abundant clonotypes. But if the hardest case when you have poor and monoclonal, so our conclusion though was that RNA-seq is not a proper technology to capture TCR repertoire in the T-cell poor polyclonal, right? Not at all. So if you have those samples, perhaps don't even try to run the tools because you don't have just enough reads. So we also talk about can you estimate the clonal diversity accurately, right? As the way we define that as a Shannon entropy, which takes the clonotypes and the relative abundance. So what you can see, there is some differences in terms of monoclonal and polyclonal. When monoclonal, it's easier to estimate the clonal diversity, just because all you need to do more or less is estimate accurately the major clonotype, right? And that's already good enough to get some idea of the Shannon entropy. As when you have polyclonal repertoire, you first need to capture all the clonotypes and then estimate relative abundance of everyone to get a decent result. So that's harder. As you can see here, the results deviates more. And actually, RNA-seq always, I think, underestimates the diversity because we don't have enough signal in the data. Okay, so to be on time, which I hope I am. So just to wrap it up, so RNA-seq profiling, we think RNA-seq-based profiling methods are able to capture the TCR clonotype and accurately estimate the diversity of the repertoire in the T-cell rich monoclonal repertoires. We should be careful when we go to the poor polyclonal samples and perhaps don't trust the results of RNA-seq. And I think the advantage of RNA-seq that the methods can do both alpha and beta chain, right? So here we talk about the beta chain, all the results were in the beta chain. However, in theory, RNA-seq has both chains equally abundant, typically. Unfortunately, the gamma and delta chains are very rare comparable to alpha and beta, so perhaps will be not enough reads. But alpha, that's one advantage of RNA-seq, that you can do both chains at the same time. So the way we see that design, especially in the cancer sequencing, it's become routine that we're sequencing RNA-seq in the clinical settings. And then we think that step number one should be utilizing the data RNA-seq to extract the TCR clonotype. And only for the tissues like poor polyclonal, we should do additional TCR-seq sequencing. This way we can increase the size, right? Like taking part of RNA-seq and combining that TCR-seq. So we think that will be feasible design. Okay, so that's work of many people in my lab, so particularly Kari, who is a graduate student. So we have postdoc openings in the lab, so if you're interested to work on the project like that, or develop new tools, or perform benchmarking, and would like to enjoy the views like that in Los Angeles, so come to work with me. So a few announcements. We're organizing another conference called ISBRA if you want to go to Haifa and present your work and spend Thanksgiving in Israel, so that's the conference for you. And I think that's the line I was recently putting in all my presentations. So we dedicate our research to all freedom-loving people around the world and the people of Ukraine who fight for freedom. Thank you.
Okay, first question there.
Thank you, very nice for the talk and for the note. So actually in my lab we have been using RNA data for the phenotypes, and one of the things that you have mentioned a little bit about it is that we found very little agreement in the phenotypes between different patients that were having the same type of condition.
Mm-hmm.
You are now saying that you have to be very careful with type of sample that you choose. So how could we measure when we can use basic data for this job?
Sure. Yeah, that's a great question. I think one part of the disagreement, you're saying you don't have shared clonotype between the samples, right? Like, but different patients, right? Like a different individual.
Yeah, different patient with the same condition.
Sure.
With the same.
Sure. So I think one reason, very simple reason, you might not be like sampling them enough, right? RNA-seq doesn't give you the depth. And actually, even big question is, will TCR-seq give you enough depth, right? Because TCR like doesn't capture all 10 into the power 15 different clonotypes, but several millions of those, right? So it's still missing the depth, right? If those clonotypes shared between the patients are rare, perhaps you really need to do deep TCR sequencing, right? So in terms of another question, when we should use, I think the key element is the number of T cells in the sample, right? Because fractionally, when you sequence RNA-seq, you're sequencing normal transcriptome and the genomes or whatever recombined genomes of the T cells. So if you have little T cells in your sample, you will not have the reads, and that's like hopeless for bioinformatics. So I think one way to know, just to use common knowledge where you would expect large number of T cells, or actually better, do deconvolution, right? Take the gene expression data, and from the gene expression data, estimate the T cell fraction in your sample. And actually, this is what one additional work we are doing right now. So instead of just assuming the skin will have very little T cells, we can actually estimate that from gene expression data.
And then you will say you need at least this percentage.
Right, yeah. So like actually, we want to put that information. You know, like if T cells fraction, it should be this in order to capture this amount of TCR repertoire, right? So we want to put that recommendation. So that will be precise in terms. Yeah, thank you.
This may be kind of a basic question, but is there an argument for doing single-cell RNA-seq to like specifically to pull out the T cells directly, and then are you going to have more power to discover this repertoire?
Yeah, no, I think it will be great because exactly you can combine alpha and beta chain, right? At least, and maybe gamma and delta. So you will have, you know, like the entire repertoire, like the entire clonotype, not just fractional one from alpha chain, but both. So I think one limitation is and I think there are technologies doing that, but you need to enrich for the T cell, right? Like normal single cell, we didn't try, but I would expect there is like given the throughput, right, will be worse than RNA-seq, will be less reads coming from the T cell repertoire. However, if you do like 10x style of enrichment of the T cell, so I think it's a very feasible technology and very promising one. And perhaps, I think it's an interesting open question, can it replace TCR-seq, right? Should we just go single cell all the time? Right, so I think that's another idea for another benchmarking, I guess.
And then like a quick biology question. Obviously, like blood, with those you expect high T cell, but when you're talking about like T cell low tissues, what's an example of one where you would expect kind of monoclonal population versus?
Yeah. So I think in our case, the monoclonal were only in the PBMC. So and then some. I think it's about the immune response, I would say. So there is a strong immune response proliferating a certain clonotype, right? So that will be abundant. So I think it should be some disease condition, I imagine, not like a healthy individual, healthy sample, rather like either engineered ones, right? Like where you engineer that, or perhaps like some severe condition where there is a strong immune response proliferating a certain clonotype.
Yeah, I have a question. I don't know if it's going to hear me, maybe you have to repeat it.
Okay.
But when you show first of all, you didn't tell us which tool is better. Do you have a benchmarking? You know, I was thinking in some of these, whether there is some metric that can tell us.
Yeah, so no, that's a great question. So the question was like about relative performance of the tools. So yeah, so that's actually work in progress. So yeah, because it's a little bit hard to do because one tool is ours and there are multiple metrics. So I think that's one challenge. No, but I think, you know, like I think we would like, I mean, there are fundamentally few questions, right? Like for example, that can be converted, like if you can look how the blue color and the orange color overlaps the y-axis, right? So that's the number. That's a fraction. So we can kind of like make a table or something to indicate what is the number, even though it's very similar, but perhaps somebody is doing better, right? And the same, so there are a few measures, which I think is a good idea kind of to summarize that. So fraction of the repertoire being captured, you know, like ability to estimate the clonality accurately, actually ability to accurately estimate the relative abundance of each clone. So there are several parameters where there are numbers. So they're actually quite similar often. So I think our conclusion will be there is no single tool which is what we're observing right now. There is not a single tool performing the like superior across all the metrics. However, per metric we have a winner, right? And perhaps that will be the recommendation of the paper, that depends what you would like to get or how you would like to characterize the TCR repertoire. You need to use that tool or perhaps the combination of the tools. That's, I think, this is what we're going to recommend.